So today I'm going to be talking very much from the perspective of a plant geneticist, which is the background I came to phenotyping from. Now I know within the cluster that you have also many computer scientists, engineers, mathematicians, and so I want to emphasize the work that I'm going to present is only possible because of some wonderful collaborations I have, including with a statistician, Yumo Chu, and an engineer, Yufeng Ga. And this is one of the really powerful and exciting things I like about working in phenotyping is that it does allow and force us to work with people with expertise very different from our own, which is a really fun feeling. Now, as many of you are already no doubt familiar, we have a real problem in agriculture, which is that we need to meet growing demand for food. We need to do it with ideally the same amount of land and ideally while reducing inputs so we can reduce the agricultural footprint on the environment. And one of the challenges we face in that is that if you look around the world, there are three major crops, rice, wheat, and maize, that are responsible for about half the calories consumed around the world. And for two of those crops in many of the highest yielding parts of the world, yields have stopped increasing. So if you look at the production of rice from a hectare or wheat from a hectare, we're not getting better anymore. And as a result of that, what we see is that the total area on the globe devoted to crop cultivation has started to increase since 2003 after being flat for many years, which is very unfortunate because the land that's coming into production is wild land that we'd really rather not be converted to agriculture. It tends to degrade rapidly over time, and once we convert it to agriculture, we can never get it back. And the reason that this total area devoted to crops had remained flat for so long was because we were able to increase yield. Now, if we look at the yield of maize, we see that the maize yields are continuing to increase year over year. So this is in US units, so this is bushels per acre, but really it's surprisingly linear. About every year we are able to increase the yield of maize by about 1.8 bushels per acre. But we are only able to do this by devoting exponentially more money to plant breeding and plant biotechnology. And so anytime you're getting a linear response from exponential inputs, that's obviously something that is not sustainable. Now, in order to increase the rate of genetic gain here in plant breeding, without continuing to invest more and more of our total resources as a society into it, what we need to do is identify what are the bottlenecks and work to open those bottlenecks up. And we've as a society, we've done this successfully several times. So initially, the major bottleneck in plant breeding was simply breeding cycles. So you need to be able to cross things together, generate new genetic diversity, evaluate those, pick the best lines, and move on. And that process takes a number of years. Through statistical genetic approaches, such as genomic prediction and selection, we've really widened up how much of that selection we can do and how fast we can do it. This created a new bottleneck, which is that genomic prediction requires lots of information on the genotype of many, many individuals. And so new genotyping technologies were developed. And again, that bottleneck widened up. So right now, the real limit on our ability to develop new, better crops, as well as to understand the functions of genes within those crops, or develop new and improved agronomic practices that will use resources more efficiently, is simply field trials and the ability to measure how plants perform in the field. So on this slide, I don't know if you can see my cursor or not, but this image on the left-hand side is probably one of the most overused slides that you will run into in the field of biology, which simply shows how rapidly the cost of sequencing has declined in our ability to measure the genotype of an organism. So whether we are doing plant breeding, whether we're trying to link the functions of a particular gene to a particular phenotype, we need both genetic and phenotypic information. The genetic information has become much, much cheaper. The cost of measuring traits of plants has stayed the same or even increased over the last 15 years, in large part because this tends to be a very labor-intensive process, and the cost of labor increases faster than the overall rate of inflation. Hence the interest in high-throughput phenotyping, which I think is still a relatively poorly defined field. We define it by what it is not. So it is not sending a combine through a field to measure biomass or yield. It's also not sending students out into a field with rulers and notebooks to make measurements, which is how the vast majority of data in my field is still collected. The challenge we face is there is a limited number of students who are willing to go out into a hot cornfield in the middle of the summer with steam coming out of the mud. And we really can't grow that supply. So we want to use that given supply of labor much more efficiently, because otherwise we can't grow the size of our data sets. So the framework I'll be using through the balance of my talk, I'm going to talk through a couple of stories on different layers of this, is what I call the Phenotyper's pyramid. And the reason I think it's useful to think of this as a pyramid is that each layer of this process depends on information from the layer below, but each layer requires a different set of expertise and techniques, which requires these collaborations across different fields. The other challenge is that it's very hard to develop the techniques and tools and methods for any layer of the pyramid without already having information from the layer below, which is really hard when you're trying to develop and scale up large new technologies in a fixed amount of time, because you'll troubleshoot one layer, then the next, then the next. So I'm not going to talk a lot about the engineering that goes into the collection of the raw sensor data, which is, I think, one of the key steps here, simply because I'm not an expert compared to a lot of you, and I would probably butcher it. But I will say we've benefited so much from this collaboration with Professor Ga, and I'm going to reference a number of his publications. So at the University of Nebraska-Lincoln, we have access to two systems, sort of large infrastructure systems. One uses a camera payload suspended from a series of cables that moves back and forth across a field. This is used for a lot of the same data that might be collected from a UAV. The advantage of this system is that we can provide a lot more power. We can carry heavier instruments. And unlike UAVs, there's not the same risk of a crash, which means we can send up really expensive instruments. When I talk to Professor Ga and say I'd like to send up a hyperspectral, a $50,000 hyperspectral camera in a drone, he starts to look pretty worried. So it's much better if we can use this system. The second system I'll talk about, I'm sure that you've run into a lot of these throughout the world, is simply an automated phenotyping greenhouse. Plants are on conveyor belts. They can move around the facility, go through a series of imaging chambers. Nebraska distinguishes ourselves in that we have one of the few systems that can grow relatively large plants, up to about 2.3 meters, which means we're able to record the entire life cycle of many grain sorghums, as well as some maize lines, though a lot of maize hybrids are vigorous enough. They still hit the maximum height on the system. So with this greenhouse system, this collects a number of different image types, but we're really going to focus on simple RGB photos taken of the plant from different angles, as well as hyperspectral photos, which capture many, many different wavelengths of light and can distinguish things that would look identical to your eye or mine, because we have only 3 sets of cones in our eye to distinguish different colors, and this can distinguish many things that would stimulate those, many different wavelengths of light that would stimulate those cones equally. So the first step where my lab really has a large contribution, rather than depending on working with excellent collaborators, is at the image processing stage. And this again gets back to this problem, which is that there are 2 ways to build a pyramid. One is you build the bottom layer, you make sure the bottom layer is solid, then you build the next layer on top of that and the next layer on top of that. And that works really well, but it takes a very long time. Particularly when you're using different groups of people to build each layer of the pyramid. So you build the first layer, you send all those people away, you bring in new people, they have to figure out how to build a pyramid, send those people away, and so forth. So the argument that I make whenever I talk to any group doing phenotyping is that it's much better to build from the inside out, where you build a little bit of the bottom layer, which allows you to build a little bit of a layer on top. And this allows us to go much faster. And so for this image data that we were capturing from the greenhouse, the facility was being built. We didn't have any data yet, but we knew we wanted to do image processing. And so we developed some low throughput solutions that allowed us to generate the same type of data that we were ultimately going to get from the facility. What you can see here, this is Jikai Liang, who was my first PhD student. This is Saram and Bhushit, 2 computer science master's students. And rather than bringing the plant in on a conveyor belt, what Jikai does is he picks it up from the bench, he puts it down on this little cart, and then he rotates it by 36 degrees between each photo. This is not a tool that I would use for scientific discovery, but it allowed us to generate the same type of data and start learning how to process it before we had the big and expensive data sets to work with. And so I'm going to tell you one of the early lessons we learned from just these very simple data sets and applying simple and quite naive approaches to measuring things like plant height and total area. So when Jakai got this early data, he collected ground truth. He did the same things probably a lot of you would do, which is that he tried to estimate, so how well can we measure biomass just from looking at the number of plant pixels in the image. It turned out we could measure reasonably well. It was an R of about 0.9, which means about 80% of the variation was explained just from looking at plant pixels. Now, if that was random error, that would be fine. We could collect lots of measurements and we would be able to accurately estimate the biomass of different individuals and also estimate the biomass of different genotypes and look at genes controlling biomass accumulation. The question Jakai asked, though, which was a very good one, is this error really random? And it turns out that it is not. It was systematic. So more than 50% of the total error with respect to biomass estimation is explained by genotype. So some varieties had their biomass systematically underestimated and others had it systematically overestimated. Once you're presented with this, it makes a lot of sense. If we're just counting total pixel number, a stem will probably be a lot denser than a leaf, but a leaf will, in a per pixel unit. You could also envision differences with leaf architecture that would affect how many pixels are obscured, and all of these are controlled by genetics. So if we had used this naive data, gone on and attempted to do mapping and link measurements from this high throughput phenotyping to genes, we would be mapping not just genes controlling biomass accumulation, but also the genes controlling error in the estimate of biomass from RGB images. And so that's a lesson we've kept in mind as we move on to more complex analyses. So as I mentioned, one of the problems there is that stem has very different biomass from leaf. So what we wanted to do is train a computer to actually separate the stems and the leaves. We did that using an online crowdsourcing platform called Zooniverse, where we put up many of these single bands from hyperspectral photos, asked people to select pixels that were either part of the stalk, part of the leaf, part of the panicle, the flowering structure, or just background. A PhD student in my lab and an undergraduate worked together to identify, okay, we have lots of differences in the reflectance patterns of these 4 classes, and train a simple algorithm to classify each pixel into a leaf, stem, panicle, or background. And what I really like about this is that even the errors make sense. So you can see there are a couple of these orange pixels in the middle of leaves showing up. And what that's doing is it's detecting the thickened midrib of the sorghum leaf, which tends to be thick and lignified and in a lot of ways is going to share properties with a stem rather than a leaf. And this is exciting because it allows us to measure, both to treat different organs of plants differently, which is, I think, quite important in minimizing error, but also to measure more relevant plant traits. So for example, the easy way to measure height that Jakai used in his very first paper is to look at the total distance between the top and the bottom plant pixel. And what you see is it's not monotonically increasing. Depending on the orientation of the top leaf of the plant, which changes over development, height's going up and down. So if you are a biologist in the field and you're walking around with your ruler and your notebook, you do not measure this height, you measure to some other landmark on the plant. And yes, as you can see, once we can do this separation of the different organs, it is possible to measure. So plant height 1 is the standard many biologists use to measure height, which is to the top of the flag leaf. You can also measure height to the base of or top of the panicle, as well as the height that could be measured with RGB images. So as we move into quantitative genetics, though, from these measurements, we could use the existing tools for conducting, say, a genome-wide association study. But high-throughput phenotyping enables, the data collected by high-throughput phenotyping has a number of distinct properties. So one is that it's a lot easier to generate data that is time series rather than single time point. I saw that one of the papers coming from PhenoRope was actually looking at 4D reconstruction, which again is, I'm assuming, having that extra dimension be time. But also that with a single piece of sensor data, whether that is an RGB image, a hyperspectral photo, a LiDAR point cloud, it is often possible, and in fact a good idea, to measure multiple distinct properties of a plant. So high throughput phenotyping data sets tend to be inherently multidimensional or high dimensional compared to the data when you have students running around the field with the rulers and the notebooks. So first, I'm going to talk a little bit about what we can do with the time series data. We'll be using an association panel of about 400 diverse sorghum lines, which have been genotyped for hundreds of thousands of markers. So the nice thing in this population is we have some known true positives. So there are some genes that control plant height in this population that have been cloned. These genes, however, cannot be detected once you eliminate the extreme lines that cannot be grown in greenhouse conditions. And in both plant biology and plant breeding, it is very common to be working with these phenotypically constrained populations. So we know where the 3 genes are. We aren't able to identify them in a conventional genome-wide association analysis. Folks who haven't seen these before, this is a Manhattan plot. So each of these points represents one marker, and the height on the y-axis indicates how significantly that marker is associated with variation in the phenotype, in this case, plant height. Some of these guys get really extreme. I think this is a photo of 2 students measuring one of the sorghum plants that is over 3 meters. This might be the 4-meter line. With the hyperspectral phenotyping, as I showed you a couple of slides before, it's possible for us to measure plant height as we would measure it if we sent a student into the field to measure it every couple of days. When we look at those heights, we see a relatively messy set of growth curves. Oh, this isn't going to play, is it? All right, we're just going to skip through that. But when we instead align by looking at a different developmental landmark, instead of looking at days after planting, we'll look at when the panicle emerges. We're able to get much more comparable curves. That's not going to play either. So simply by measuring all of the time points, instead of identifying 0 of the 3 genes, we're now able to identify 2 of the 3 genes. So what you're seeing here is the same type of Manhattan plot you see here. It's simply been flipped so that this y-axis corresponds to this x-axis, and then each column represents an analysis conducted at a different time point. If any of these points are above the line of statistical significance after correcting for multiple testing, you'll see a colored band. So we can see some things are detected over many days, some things are detected rarely, and there's no signal left by the end of the experiment. We are also concerned in doing this alignment, this would instead introduce error produced by identifying genes controlling the timing of reproductive development. Something that flowers later will probably be taller than something that flowers earlier, but we did not see significant overlap between the genes controlling timing of flowering and inflorescence development. However, we're still identifying only 2 of the 3. The other challenge is that there are a lot of signals here. And if someone were determining, okay, which of these signals am I going to invest a lot of student time in mapping and characterizing a gene, characterizing any one of these is probably 3 to 5 years of a plant biology PhD student's life. So we want really strong evidence. This isn't a very clean argument for that. So this is where cool statistical techniques come in. So we worked with a statistician named Yuhang Xu, who was able to use non-parametric functional principal component analysis. So taking all of these curves, calculating an average of these curves, and many more, there were about 300 total genotypes analyzed, and calculating 2 functional principal components, which explain how individual curves vary from the average curve of all the plants. And so for, you can see PC1 essentially corresponds to either being taller or shorter than the mean, while PC2 corresponds to being taller prior to this upward slope during bolting and panicle emergence, but shorter after that point. And so with these principal components, we now have a score for each principal component for each of the individual plants. and we can again conduct a genome-wide association analysis, and now we get a much cleaner result where we are able to identify all three of our known true positive genes, a couple of new signals that we might want to go after. But what I also like is this actually provides a little bit more insight into how these different genes influence sorghum height, with dwarf 1 and dwarf 2 influencing height throughout development, and dwarf 3 primarily influencing height prior to flowering, which after we found this, we were able to go back into the literature and confirm that this is what is known about how these two genes influence plant development. So the second difference I brought up about high-throughput plant phenotyping is that it is inherently high-dimensional. And this is not something unique to high-throughput phenotyping of plants. People are seeing the same thing with, for example, mining medical records, where you can get many, many measurements of different properties of humans, or working with molecular traits. So if you conduct RNA sequencing, you'll measure the expression of tens of thousands of genes, and you can measure that across all populations, or scoring different types of metabolite abundance. For this, I'm going to be working instead of sorghum with a maize population, slightly smaller with about 280 lines, but genotyped for tens of millions of markers. And by mining the literature, we were able to assemble a set of about 260 unique phenotypic data sets, which can either be different traits or the same traits scored in different environments in different years. You can see that some of these phenotypes are quite correlated with each other, others are not. If we use conventional genome-wide association analysis, in many cases, no single gene trait association will be significant, because you have cases like this particular gene. This is a slightly different visualization, so the y-axis still indicates statistical significance, but now the x-axis is indicating each of the different phenotypes, and we've clustered them so that phenotypes that are correlated appear next to each other. So in this particular case, the gene was not significant in any one phenotypic data set, even though it came close to it in many. Anyway. So what Jikai Liang did in his last project in my lab was to work with a statistician to develop a genome-wide, phenome-wide association study that uses both multiple SNPs, multiple genetic variants present within a gene, but also large sets of phenotypes which do not need to be correlated with each other, and is testing for essentially the association between that gene and phenotypic variation of any sort, and then it identifies a set of phenotypes associated with that genetic variation. So for this particular gene, where it was not, no one phenotype was significant, when you conduct a combined analysis using both multiple SNPs and many traits, we identify sets of phenotypes that are selected, and the overall association is significant. Now, I just deleted about 4 slides out of this talk where I talk in detail about the method. If you're interested, those are in the paper, but in order of finishing this, not going too late into the hour, I'm not going to go into the methods here, but let me know if you have questions. With what's documented online, send me an email. So we can try and validate how well this is working quantitatively, looking at sets of genes that were identified in a different analysis of a much larger and better powered population, and say that our overlap and our validation rate is substantially higher. It's significantly different from many methods that are single trait. For me, I think this is the more convincing statistical argument, but the more compelling argument is to look at individual cases. So this is Anther-ER1. It's one of the classical mutants of maize. It was not identified in any conventional genome-wide association of this using this data set. It was identified through the genome-wide, phenome-wide association analysis. And we can go into the literature and identify the different phenotypes that are associated with the loss of function gene, and compare those with the phenotypes that are identified by this GPWAS model, simply by mining all of the trait data that's been identified for this population. When we do that, we see that many of these phenotypes line up very nicely with the mutant phenotype for how when this gene was studied in the past. or with other quantitative genetic studies using larger and more highly powered populations. This is another example of the same thing, where the model identified a known classical mutant ligulus 2 as associated with phenotypic variation in the population, natural variation, and then if we go and look at the loss of function phenotype, many of the phenotypes identified by the model are associated with the phenotypes from the loss of function mutant. So at we've sort of climbed this model, touching very briefly on engineering, then going to image processing, from image processing to quantitative genetics. But then from quantitative genetics, we really do want to understand something new about the biology of the organism. And so when we looked at the set of genes identified through this genome-wide phenome-wide association, it was only about 1,600 of the 32,000 genes in maize. And we wondered these genes might be different from other genes in the genome in some way. When we started asking this question, it then became apparent to us we had to define, well, what's a real gene? What is a mistaken annotation on the genome? And this sounds like it should be a very simple question, but if you go to Google Scholar and you search for what is a gene, you will find a review like this one, which, okay, well, this entirely loses its effect without animations. But a very complicated definition. As mentioned at the start of the talk, I'm also involved in a couple of companies there. We have to deal a lot with lawyers. Maybe it is different in Germany. In the US, the legal process is incredibly complex. And what I would say about this definition, this is a definition a lawyer could be happy with. It's not a very helpful definition when you're trying to distinguish true positives and false positives. So instead, if you dig into the literature, you try to find the origin of the word gene. You come to a Danish botanist named Wilhelm Johansen, who published originally in Danish, which I do not read. His book was then translated into German, which I read extremely poorly, but I read enough to find the right sentence to plug into Google Translate, who defines a gene as simply the special separable characteristics of an organism that condition, or features of an organism that condition the characteristics of that organism. So a gene is the thing that causes phenotypic variation. If we look at those classical mutants I was showing you previous slides, so this is something where someone has broken the gene and shown that there actually is a change in the phenotype of the organism. There aren't a huge number of those. There are a couple hundred in maize, maybe 2,000 in Arabidopsis. But they have some different features. So one is they're much more likely to be conserved at the same location in the genome between species. They show different patterns of methylation. They are more likely to be transcribed from DNA into RNA, and those RNAs are more likely to be translated to protein, which again makes sense. While there are some exceptions, usually it's very hard for a piece of DNA to affect the phenotype of an organism without being made into a protein. And in fact, we can separate those from the overall set of gene models in the genome. So if you look at only those genes where the gene has been disrupted, there's a loss of function phenotype. So breaking the gene actually affects some known characteristic of the organism. They're separated on a large number of factors, which I won't go into in great detail beyond what I've just told you. But when we look at these genes identified by GPWAS, so genes associated with natural variation in phenotype in large diverse populations, they exhibit many of the same features. They tend to be more broadly expressed across tissues, more conserved in the same locations between species. They're less likely to exhibit presence-absence variation. They also just tend to be longer than the average gene model. And this goes through that in a bit more detail. But this got us quite excited because, you know, if classical mutants are different from if these loss of function mutants are different from everything else, there could be a lot of explanations for that. But if we see a common signature, both for the classical mutants identified by geneticists over the last century and the genes affecting natural variation in breeding populations, that suggests that it should be possible to train prediction models to distinguish genes that will have gene models that will have phenotypic effects from the overall set of about 30 30 to 60,000 genes in the maize genome, depending on who you ask. So that's what another student in my lab, Shiru Dai, did, is she assembled a large number of different descriptors, features of gene models in the genome, and used random forest prediction algorithms to get surprisingly accurate prediction of which genes would produce mutant phenotypes and which genes would be associated with quantitative phenotypic variation, and is currently working on how do we merge those models together so we can get even better predictions. Right now, we're not quite there yet. So starting with a collaboration with engineers to collect image data of plants over time, we were able to develop methods to extract measurements from those images, develop new quantitative genetic techniques to take advantage of both multi-time point phenotyping as well as high multi-dimensional phenotyping. And from that, develop a model that will aid reverse genetics, because if we can do better prediction of which genes it makes sense to go after, this significantly improves the productivity of genetics, where characterizing a gene takes, again, 3 to 5 years, starting from scratch. And right now, what often happens is the student spends 3 or 4 or 5 years and cannot identify any phenotypic outcome at all. So being able to predict that both increases scientific productivity. I think it also increases biology graduate student happiness a lot, which is a worthy goal. So with that, I think I'm right around 40 minutes. So thank you all again for this opportunity to talk. And while I've tried to highlight some of the collaborators that contributed to this work, I have everyone listed on the slide. Thank you.